Okay, now the topic of my presentation is a sustainable approach for the green and efficient extraction of carotenoids from Rhodotorula GLUTINIS. In this concept, I will talk about carotenoids, Rhodotorula GLUTINIS, hydrophobic deep eutectic solvents, and then I will give information about materials and methods. Then I will give you results and conclude the study. Oxidative stress is the main risk of chronic diseases. To prevent this, we need to consume fruits and vegetables, which are good sources of antioxidant phytochemicals. One of the major class of antioxidant phytochemicals are carotenoids. Carotenoids are fat-soluble molecules, and they give different colors to the foods, and they play an important role in the prevention of human diseases. They are antioxidant molecules, and they can act as vitamin A precursors. They are synthesized by plants and microorganisms, but not animals. So they receive considerable interest by researchers. Industrial production of carotenoids is made by extraction from plants or chemical synthesis. Chemical synthesis is preferred due to its cost-effective production than biotechnological processes. What about microbial production? Of course, there are many advantages related with microbial production because it requires a small production area and it's independent of changes in climate, seasonality, and also soil composition. But for the microbial production to be industrially viable, costs should be minimized. One of the ways to manage this is to use the side streams of the food and agriculture industry as low-cost sources as nutrients. The traditional recovery of microbial carotenoids are made by using non-polar volatile organic components for highly hydrophobic carotenoids and also slightly polar volatile organic components for less hydrophobic carotenoids. Olive meal wastewater is a byproduct of olive oil production process. It has a dark color, low pH, it has a strong smell, and its waste amount reaches to almost 30 million cubic meter per year, which is a huge amount. But it contains phenols, sugars, lipids, and polyalcohols inside. And Rhodotorula GLUTINIS has the ability to use this waste as the growth media. Rhodotorula GLUTINIS is a pigmented aerobic yeast. It's important for food industries because it has biotechnological potential and it's a safe microorganism. Rhodotorula GLUTINIS is capable of producing carotenoids during the stationary growth phase. But the carotenoids are intracellular molecules, therefore, their recovery requires a series of downstream operation units. And the extraction step from microorganism is critical to recover the carotenoids. Rhodotorula GLUTINIS mainly produces three components of carotenoids. These are taurularhodin, taurulan, and beta-carotene. We can see their structure in the slide. Among these components, taurularhodin is only synthesized by microbial species, particularly by the genus Rhodotorula. Deep eutectic solvents are new generation green solvents. They are prepared from two or more components. At least one of them should be hydrogen bond acceptor and the other should be hydrogen bond donor. Their advantages are they are low-cost solvents, they are biodegradable, they can be easily prepared, they are sustainable and environmentally friendly solvents. Hydrophobic deep eutectic solvents are presented to the literature in 2015 by Van Osch and friends. The difference of hydrophobic deep eutectic solvents is the hydrophobic nature of these solvents. Their advantage is the same, their eutectic property is the same, but these are composed of only hydrophobic molecules. Our aim in this study is to present sustainable approach for green and efficient extraction of carotenoids from Rhodotorula glutinis. For this aim, we use olive meal wastewater as a zero cost source for the cultivation of the microorganism. Then we used green solvents, which are hydrophobic deep eutectic solvents in the extraction of carotenoids. Thereby, we eliminated toxic organic solvent utilization, and we use an efficient method as ultrasonic homogenizer to be able to easily disrupt the cell, which has a rigid wall structure. For this, we used olive meal wastewater. We centrifuged the waste and used the supernatant. We prepared universal yeast medium using olive meal wastewater, and then we inoculated the cells into olive meal wastewater containing 1 gram per liter urea. And we fermented for 144 hours at 30 degrees and 150 RPM. After that, we centrifuged and lyophilized the cells. After collecting the cells, we prepared 4 different hydrophobic deep eutectic solvents. These were menthol-based deep eutectic solvents, and the components were acetic acid, propanol, and butanol. We used ultrasonic extraction. We put the cells and hydrophobic deep eutectic solvents, and we sonicated for 10 minutes. And then we centrifuged the cells, and we used the supernatant to calculate carotenoid content and also carotenoid profile with HPLC. Then we performed the experimental design using 2-factor central composite design. The factors were chosen as solid to liquid ratio and extraction time, and we can see low and high values of the factors. Here we can see 13 different data sets of the experimental design. According to the results, we tested different percentages of olive meal wastewater in the growth of Rhodotorula glutinis, and we found out that the highest cell concentration could be obtained in undiluted olive meal wastewater. So we produced the cells in undiluted olive meal wastewater. After extraction, first of all, we screened traditional organic solvents in the extraction. For this aim, we used ethanol, acetone, methanol, chloroform, and hexane. And the highest total carotenoid content could be obtained using methanol as 77.35 microgram per gram cell. Then we screened different hydrophobic deep eutectic solvents. Here we can see that all of the deep eutectic solvents provided higher results than reference methanol. On the other hand, the highest carotenoid content could be obtained using menthol acetic acid deep eutectic solvents, giving 157.13 microgram per gram cell carotenoid content, which is almost twofold higher than the reference solvents extraction. Here we can see the effect of hydrophobic deep eutectic solvent type on carotenoid profile. The numbers at the bottom shows us total carotenoid contents obtained with the hydrophobic deep eutectic solvents. Different color bars shows us torularhodin, torulen, and beta-carotene contents. We can see that the highest carotenoid component was obtained as torulen for each used deep eutectic solvent. And the composition of menthol acetic acid extract was found as 49% of torulen, 22% of beta-carotene, and 17% of torularhodin. ANOVA results show that the response was expressed best with the quadratic model. Lack of fit was not significant, and we obtained a high value of R square values. We can see the equation obtained for carotenoid content in terms of coded factors in the slide. Here we can see the response surface graph. This graph shows us that solid to liquid ratio, as solid to liquid ratio increased, carotenoid content also increased. And as time increased, carotenoid content increased, but then almost remained constant. Then the program gave us the optimum extraction conditions as 0.02 gram per milliliter of solid to liquid ratio and the extraction time as 13.16 minutes. When we performed this, experimental response was obtained as 239.27 microgram per gram, which was very close to the predicted response. So optimization was accomplished successfully. In the literature, there are only a limited number of studies related with microbial carotenoid extraction using deep eutectic solvents from different sources. Therefore, here to present, we chose 3 example studies. These studies presented carotenoid extraction from Rhodotorula genus with novel systems or solvents. The first study was performed by Musagi and friends. They used Rhodotorula GLUTINIS. They used different ammonium-based protic ionic liquids for the extraction of carotenoids. For the extraction strategy, they used mixing. They reported that as temperature increased and ionic liquid level percentage increased in the extraction medium, they could recover higher amounts of carotenoids. And the best solvent is reported as DEPA-hex solvent. They obtained highest carotenoid component as beta-carotenoid. And here you can see the carotenoid composition presented by the researchers. Another study was performed by Martinez and friends. They used again Rhodotorula GLUTINIS for the extraction, but they used a novel strategy. They used ultrasound under pressure with phosphate, citric acid and disodium hydrogen phosphate buffer. And they presented the best results, best extraction parameters as 200 kilopascal and 120 seconds. Under these conditions, they obtained 220 microgram per gram total carotenoid. The other study is performed by Costa and friends. They used Rhodotorula mucilaginosa as the microorganism, and they used different deep eutectic solvent types, but they are choline chloride-based deep eutectic solvents, which are hydrophilic deep eutectic solvents. They used vortex at 50 degrees for 1 hour, and they presented best deep eutectic solvent as choline chloride glycerol mixed with 30% of ethanol in the extraction medium. And they reported that they obtained 110 microgram per milliliter of total carotenoid in the extraction. If we summarize reference studies, we can see that they use tailored media for maximum carotenoid production, and they reported carotenoid content between 200 and 400 microgram per gram. In the present study, we used a byproduct of olive oil production, olive meal wastewater as the growth media. And we used hydrophobic deep eutectic solvents as extraction media. And the highest carotenoid amount was obtained as 239 microgram per gram, which was comparable with the literature. Here we presented first-time use of hydrophobic deep eutectic solvents in the extraction of carotenoids from Rhodotorula GLUTINIS. And moreover, these components are natural components. Therefore, they don't need any purification, any further purification. So this extract can be directly used in cosmetic field or food industry. So this has another advantage of using natural components in the study. We are continuing to making experiments related with this subject, and further studies may also include utilization of different waste as growth media for Rhodotorula GLUTINIS, and cost-effective additives can be added to the media in order to enhance carotenoid production. And of course, there are many different types of hydrophobic deep eutectic solvents, so they can be tested as extraction media. Here are our references. Thank you for listening, and I have special thanks for Abdulkadir Keskin, who performed these experiments as a PhD student. So, do you have any questions to me?